All right, welcome back everybody. We're going to get started with our next lab, the Integumentary System, and start a brief introduction into our skeletal system. So up here, as you can see, we are going to be talking about our skin. We've got some interesting folks down here at the bottom of our screens. And we are going to be talking about where some of this ink on these two guys' skin is going to actually be going. We'll be talking about what's happening when you're laying in your tanning beds. Hopefully you actually close them. From what I hear, you get a much better tan that way. So let's start with a handful of review questions, just to be thinking about last week's lab and kind of bring that into today. So take a second, and which of the four types of tissue is the epidermis composed of? All right, hopefully you said epithelial tissue on that. And so which specific type of epithelial tissue then is the epidermis composed of? So think about all that we went over last week with the different types of epithelial tissue. All right, I hope you said keratinized, stratified, squamous epithelial tissue. All right, now which of the four types of tissue is the epidermis attached to? And hopefully you said that that would be connective tissue. And then what is the specific type of connective tissue that dermal layer is made of, or our dermis? So there's actually two types that we'll see, the areolar tissue, which is our loose connective tissue, and a dense irregular connective tissue, which is a dense connective tissue. So we will be getting into that this lecture and sort of hashing out what is the difference between those. All right, so let's start talking about some of the major structures in the integumentary system. Now, first and foremost, what do we know about our skin? Our skin is the largest organ of our body. And what we'll see is that associated with our skin, we have hair. As you can see over here on the right, we have some hair follicles and some hair sticking out. We have our nails, and then we're going to be talking about these different types of glands that you can see here in purple and green, and also connected directly to our hair follicle there. So why do we have skin? What does it do for us? Have you ever appreciated your skin? Have you ever thanked your skin for the many or the multifunctional talents it gives you? So first, it helps us with thermal regulation. So thermo is temperature and regulation. We are modulating or managing our temperature in our body. And so it can help with cooling us down when we are sweating by excreting sweat, which evaporates, cools the surface down. We'll also see in today's lecture that we'll have vasoconstriction and vasodilation of blood vessels. It also protects us, so it prevents excess water coming in or losing water. It helps to reduce abrasion. It actually helps us protect ourselves from UV light. And it offers a lot of sensation, so touch, pressure, pain, and temperature. And you'll be learning a lot about these, how this works in your nervous system in lectures. And it also is involved in vitamin D synthesis in the presence of sunlight. Now, what we want to talk about are the different regions of our integumentary system. So the first broad region is our epidermis. We've mentioned that a couple times. Below that, we have our dermis, and below that, more deep to those two layers, is the hypodermis. And the hypodermis acts as an anchor between our integument and our fascia, and this is where our subcutaneous or our hypodermic injections will go, because this is highly vascularized, and so we'll be looking at that today. Now what we're going to be doing is breaking the epidermis into different layers, and we can see these layers starting superficially with our stratum corneum and moving more deep to our stratum basale. We can see then our dermis is more deep to that, and then again that hypodermis is below that. So we're going to be looking at these 5 layers in depth. This is just a nice reference image, kind of talks about the different types of cells that we'll see in those layers. So dead keratinocytes, living keratinocytes, what are they going to be doing? Our dendritic cells, tactile cells, so picking up a little bit of sensory information, melanocytes, and some of those sensory nerve endings. Remember we said with our epithelial tissue we have almost no matrix between those cells, and we're not going to have that innervation and those blood vessels coming up into that tissue. Now, a closer look at these different layers. We've got them colored in here with some lines. So out here would be our external environment, and deep to this white line is our stratum corneum. We can see that this layer is really, really thick, and if you remember just the previous slide, that is dead keratinocytes. So if we were looking in there, we wouldn't see any nuclei. Below that is our stratum lucidum here in blue. Now luci means light, and we're going to go into this layer a little bit and where we see it and where we don't. Below that is the granulosum. Down here, a thicker layer is our spinosum, and then down here at the bottom is our stratum basale, and then we can see our dermis down here. That basement membrane would be running right between our stratum basale and the dermis. And so stratum basale, remember BAS, basale for our basal cells adhering to our basement membrane. Remember stratum, all stratum means is a layer. Let's start with our deepest layer of the epidermis, our stratum basale, denoted down here as number 1, just for reference. So this is our deepest layer. Remember, the basale is adhering to that double-sided sticky tape, that basement membrane, the non-cellular adhesive layer. It's a single layer of cuboidal cells. They may even also look slightly columnar under a microscope. And again, that's where the basement membrane is attaching. Now what's going on with these cells is they are rapidly dividing, and they are pushing their daughter cells upwards into our next layer. There's 2 types of cells that you'll find here. The first are the keratinocytes. They're going to produce keratin, and that keratin is going to be really important in layer number 5, or that stratum corneum. This is going to protect against pathogens, as well as that wear and tear, just rubbing and reducing abrasion. And then we also have melanocytes, and the melanocytes are going to produce melanin, and the melanin is what gives us our skin pigments. It's only found in the epidermis, and that melanin is also going to help with UV protection. So don't confuse this with melatonin. That comes from your pineal gland in your brain and helps you fall asleep. So don't activate your melatonin while you're listening to this lecture. So a couple of interesting facts. In this basale, the keratinocytes actually phagocytize or ingest the melanin that the melanocytes are producing. And when they do so, they actually ingest them and move them to the side of the nucleus that's being exposed to UV light. And so it basically creates a little bit of a hat or an umbrella over top of where that UV light is coming in to protect the DNA. That UV radiation can damage the DNA, create mutations, and there's all kinds of problems from there. So the melanocytes and the keratinocytes work hand in hand. One of the most important pieces of information that you're going to want to remember and walk away from with this is that if you look in the bottom right corner, we have a diversity of skin tones here. And everybody in this image has about the same number of melanocytes in that stratum basale. But what is different is the rate of melanin synthesis and how spread out or concentrated those fragments become. So up in the top left, with a very fair-skinned individual, they would just have a much lower rate of melanin, and those melanin fragments would be spread out. In the bottom right, we would have a much higher melanin production and more concentrated melanin fragments within those keratinocytes. So really, it's important socially and as a country to be thinking about the fact that scientifically, everybody's got the same amount of melanocytes. All that is different is actually the coding that's coming out of some genes in the DNA, which we don't really have any control of anyways. So now moving up to our next layer, our more superficial layer, is our stratum spinosum. And this is several layers of keratinocytes thick. Basically, they continue to push forward because of the cells from the basale that are pushing upward, but they do stop dividing and they produce more keratin. That keratin actually starts to flatten out those cells. So if you remember, the basale was more of a cuboidal to columnar cell. Now what's happening is we're seeing that transition into our squamous cell shape. Moving on, our next superficial layer here is number 3. It's a little bit more dark staining, and this is our Stratum Granulosum. It gets its name from little grains, so granule. This is only about 3 to 5 layers thick, and it is even more flattened out keratinocytes. We tend to see more layers in our thicker skin, so on the palms of our hands or the soles of our feet, than in our thin skin, in our forearms or in our elbows. And so here, the keratinocytes have dark keratohyalin granules, which is where the name comes from. And basically, this is the point of where we are coating the cells, or the cells are coating themselves in this protein. And as they do so, they're exocytosing these proteins out to their surface on their plasma membrane. In doing so, they are now cutting off diffusion, or the ability to receive nutrients and get rid of waste. So in effect, they're kind of committing suicide here. So as that happens, we're waterproofing our skin and we're protecting those deeper layers and those deeper strata. So if you remember last lecture, we were looking at a zoomed out view of our stratified squamous tissue, and I said there was this really dark line, and we would come back to that. That's this line right here, our Stratum Granulosum. So again, we're going to cut off nutrient supply, so these layers of skin above it are dead, and the layers below it are alive. And you can also tell because we can see nuclei in these living cells, and we do not see any nuclei in these more superficial layers. Now our next layer is the Stratum Lucidum. This is number 4 here. And Lucy, if you remember, means light. It's a very translucent layer, and we only really see this in our thick skin. So if I were to ask you, or one of your TAs were to ask you on a test, name the layers of your thick skin or name the layers of thin skin, you would want to make sure that in your thick skin you're including the lucidum. So if you were to take a hypodermic needle and you were getting a shot or getting blood drawn, you at your elbow, you would only actually have 4 layers there instead of this 5th lucidum layer. Again, doesn't stain well, which is where the name comes from, and we see that there are no nuclei because they're dead because the nutrients have been cut off by that stratum granulosum. Okay, now more superficially, remember our basale, it's replicating, it's pushing all of these cells upwards. So this is our tougher upper layer, and it's really thick. We can see here it's the thickest of our layers, up to 30 layers of dead scaly skin cells. These basale cells that we're replicating By the time they divide and make their way up through all of these layers, about 6 weeks later, it will slough off and float around your house as dust or get stuck on your bed frame. And so basically you will expect to shed about a pound a year of dead skin cells that will flake off. Dandruff is your skin cells flaking off in your scalp, but they get clumped together with some of the sebum that's produced in the sebaceous glands up there. Now, just a quick, obviously you can make up your own mnemonic, but if you're moving from superficial to deep, you could say, can little girls or guys shoot baskets? You can make something up. You could go from deep to superficial. Here's just a real quick one to keep in the back of your mind. So take a minute, pause the video here. Would the epidermis be a good spot for a tattoo? And why or why not? Okay, so what did you say? Hopefully you said, no, it would not, because your tattoo would only last about 6 weeks. It would slough off after about 6 weeks. So we want to put that ink deeper into the dermis, which is not going to be sloughed off. We can see over here in our image that we have just talked about this part of our integumentary system, so the epidermis right here. We are now going to move deeper into our dermis. So we're going to be looking at all of these structures along the way here. So this is going to offer structural integrity. Remember, we have connective tissue here, where we had epithelial tissue in the epidermis. It's going to be made of our dense, irregular connective tissue, and that's going to be allowing for space in that matrix. Remember, we had lots of irregular collagen fibers moving in all kinds of different directions, so that's going to withstand stress in different directions. But then we had space in there. We had more matrix in connective tissue than we did cells. And so those blood vessels and nerves are able to move through that tissue in that extracellular matrix. We'll also find a lot of sensory receptors down here, giving us information about pain or heat or touch. And then we'll see that we've got our hair follicles here coming out of that dermis and protruding through our epidermis and then into the external environment. And that's going to give us some sensory information about the world as well. And again, glands that will be associated with that hair follicle and a handful of others that are going to release sweat and help with thermal regulation as they release sweat to the external surface. So let's take a little bit of a closer look. We said there were two major layers, our papillary layer, and our papillary layer is made of areolar tissue. So lots of space in there for blood vessels and nerves to pass through. Now we have these structures called dermal papillae, and basically you can think of these as little finger-like projections that are from the extensions of the dermis that push up into the epidermis. And what that does is it increases surface area for the basement membrane to adhere the two layers together, but it also forms the ridges of our fingerprints. The reticular layer is a little deeper, and this is where we said this was our dense, irregular connective tissue. And in our dermis, we'll see that we find collagen, elastic, and reticular fibers scattered throughout. So let's look at what that looks like under a microscope. So here we've got our basement membrane. We can see we've got a darker staining region here in blue, and we can see all these nuclei representing lots and lots of cells, so highly cellular, no matrix. Over here we can see some nuclei here and here and here, but lots of fibers spread out in here. And so we have more matrix than we do cells. So this would be our stratum basale, here would be that stratum granulosum, and up here would be our stratum corneum. Here is that dermal papilla. It's pushing up. It's this little finger-like projection. So if you were to take your hand and put it out in front of you and look at the back of your hand, you're going to see those fingers on your hand are pushing up. In between your fingers would be full of epithelial tissue. So here is that areolar tissue. We just talked a little bit about having that space and allowing blood vessels and nerves in there. And again, our reticular layer is going to be deeper to this tissue, and that is made of dense irregular connective tissue. And again, this is because we have those collagen fibers going in all different types of directions. So as you're pulling on your skin in different directions, it's able to withstand those forces. Now, I like to call this part of thermoregulation the Dermis Furnace, right? A furnace responds to hot and cold temperatures in your house. If you get too hot, it kicks on and you're going to cool off the house. If it gets too cold, your heater's going to kick on and warm up the house. So we said there are not blood vessels in that up into our epidermis, but they exist in our dermis, and we can see that right over here. We've got some blood vessels along the superficial portion of our dermis, and they go all the way down deep into that hypodermis. So our nutrients then will be diffusing out of those blood vessels through those capillaries into the stratum basale and migrate upwards until they reach that stratum granulosum, which is our barrier to the more superficial layers. This is going to help with heat regulation. So as our body temperatures increase, these blood vessels are going to vasodilate. So they have smooth muscle inside that's going to relax. It's going to increase blood flow. And as we increase blood flow into this upper dermis, we're going to actually release heat into the external environment, which is why if you have been near someone who's been working out, you can feel the heat coming off of their bodies. As the temperature drops, or if we were to go outside on a winter day, your blood vessels would actually vasoconstrict, and that would decrease the blood flow to the surface of your skin, and that's going to hold on to heat. If you get stuck outside and you're in the winter, you might be subjected to hypothermia because your blood vessels have essentially cut off blood flow to various structures on your body, like your nose or your ears or your fingertips, and then by cutting off and vasoconstricting these blood vessels to try to retain as much heat and keep your internal organs warm. Now, some sensory structures that we're going to need to identify. You'll be looking at these structures in labs. They are a little bit tricky to find, and so my recommendation is when you think again, if you hold your hand up in front of your face and you're looking at the back of your hand, we said that we have our dermal papillae, these finger-like projections that I'm tracing here in the blue laser pointer. So as you look at this distinction between the epidermis on the right and the dermis on the left, we have those dermal papilla. Well, again, if you look back at your hand, you see your fingernails up at the more distal portions of your fingers. And so those are similar to our Meissner's corpuscles. We find these right at the top of our dermal papillae. You can think of these as those fingernails. And so they're going to sense light touch. So up here on the superficial portion of our skin, if there's a light touch, maybe a fly lands on you, you're going to be able to feel that with these Meissner's corpuscles. Now our Pacinian corpuscles, they sort of look like an onion chopped in half, and these are going to be deeper in our dermis, and they're going to sense deeper touch. So they are pressure receptors. So if someone is poking you, you're going to be triggering these Pacinian corpuscles. Some glands that we'll be looking at, there's 4 types that are found in our integumentary system. So the first are sudoriferous glands. These are going to produce sweat. We have sebaceous glands. We can see these sebaceous glands, these flask-shaped glands associated with our hair. Ceruminous glands, these can be found in our ears, and they produce earwax. We're not going to be looking at those in this lab. And our lactiferous glands, you'll be looking at those in 202, and they are going to be producing milk. So I'm not sure which is worse, to be the subject or the scientist in this study, but we're going to first talk about our sudoriferous glands, and these are composed of our simple cuboidal epithelial tissue. So hopefully you're thinking to yourself, ooh, simple cuboidal epithelial tissue is involved in absorption and secretion, and we're talking about sweat glands. So these cells are going to be producing sweat. So we can find a couple of different types of sweat glands. The first is a merocrine gland, and the merocrine glands are simple tubes. They're temperature sensitive, and they basically release sweat through exocytosis in these little vesicles. Apocrine glands, when they are producing sweat, they actually release some organic acids because a larger part of the cell is actually breaking off into the gland. So with it, you get more organics. And so these are really close to hair follicles. And so these are involved with what's called bromhidrosis, or body odor. And when we look at up and close to this, again, our sweat is coming out. So in our armpits or our pubic regions, we're releasing these organics into the sweat, which is then getting onto the surface of our body. And, well, our bodies are covered in bacteria. So the bacteria are eating those organics, those organic acids, and it's not you actually releasing your bacteria. So the next time someone tells you that you smell funny, you can blame it on the bacteria on your body and say, whoa, whoa, it's not me, it's my bacteria. So our sebaceous glands then are going to secrete sebum, and sebum is our body's own lotion. So when we talk about lanolin and skin creams, that actually came from sheep, and so we now have more artificial forms or synthetic forms of lanolin that we put into our moisturizers. So these again are these flask-shaped glands that are right next to our hair follicles. You'll be looking at these under a microscope here. So here's that hair follicle. You can see some sebaceous glands there. And again, depending on which slide you're looking at in lab, you might see it look more like this flask-shaped, or you might see it looking something like that. So again, spend some time looking at those slides in lab as well as open labs. And so that sebum, again, helps provide waterproofing. So we have that granulosum layer, but then our bodies also distribute and secrete sebum around those hairs. So some structures you're going to need to be aware of. You'll be looking at a slide of this in the lab. So the first is our hair papilla. This is a collection of capillaries here that is going to provide nutrients to the growing hair and take away wastes. Our hair bulb then is associated with that papilla and those capillaries, and our root sheath encloses that hair. So we can see that sheath coming down, has a little bit of a bulge, and we see that this is where some stem cells are located. That's going to help with that follicle growth. We also see hair receptors or little nerves around the hairs that are going to send sensory information back to our brain so that we know that something is moving our hair. A little bit more superficially, we can see our sebaceous glands here, these flask-shaped cells that are going to secrete sebum. And over to the left, we can see an apocrine sweat gland that's going to be potentially in our armpit or, again, that pubic region, releasing sweat as well as those organics. And then right here, we have some smooth muscle called the piloerector muscles. And these are going to pull on that root sheath and potentially raise our hair up a little bit. So here is our 5 layers of our epidermis. Why don't we take a little break here? When you come back, we'll get started with our introduction to bone. Spend a little bit of time, look over the epidermis, look over the layers, draw it out, spend some time looking into your dermis, and then we'll get jumping into bones here soon. All right, everybody, welcome back. Let's start talking about our skeleton. So what does this thing do? Well, it is there for support. So if you're ever feeling down, you can lean on your skeleton. It's there to protect our internal organs. It helps us move through the world. It allows for the production of red blood cells and white blood cells. That's a really important function. It acts as a mineral reservoir, and it helps to detoxify and remove heavy metals from our blood. So we can divide our skeletal system into two major divisions. Our axial skeleton, axial, think axis, so this is our skull, our hyoid, our vertebral column, and our ribs, and our sternum. And the appendicular skeleton is everything that comes off of that. So our pectoral girdle, our upper extremity bones, so in our arms, our pelvic girdle, and then our lower extremity bones, so our legs. Now you're going to need to know all of these bones for this lab, or I should say over the next few labs. But for the course of Bio 201 lab, you will need to know all of these. So we can see in yellow here our axial skeleton. In grayish blue, we can see our appendicular skeleton off to the side. Now what's more is that you're going to actually be memorizing tons of features on all of these bones, and those features are locations that either muscles or ligaments will attach. And so those features that you are learning on these bones are really important, because when you get into the 3 labs of muscles, you'll be seeing that those features we spent time learning for the next 3 labs are actually points of attachment, insertions and origins, and you're going to need to remember those for your lab quizzes. So what is bone made of? Well, it's organic and inorganic materials. Our organic materials are cells and collagen, and our inorganic is hydroxyapatites. This is going to solidify our bone and make it really, really strong. So those minerals are going to resist compression, and the collagen fibers help to resist tension. So we can classify bones by their shape into 4 different categories. We can have long bones, so they are longer than they are wide. Short bones, which are about the same length as they are in width. Flat bones, well, they're flat or slightly curved. And then irregular bones that don't really fit into any of those categories. We can also say that there's a category called sesamoid bones, and these are bones like your kneecap that are found within tendons that can help kind of act as fulcrums. So these are the 4 major categories that we'll be looking at in these labs, which is why we don't necessarily go into the sesamoid bones. So our long bones, so examples are ulna and our radius, our femur and our thighs, our talus down in our feet, the capitate up in our wrists. Scapula is a flat bone, and our sternum is a flat bone. Our sphenoid, wow, that's a crazy looking bone, right? That's up in our skull. And our vertebrae, they don't necessarily fit in with these other groups, so they're in that sort of irregular pattern or group where kind of, scientists just dump everything else. So if we talk about a long bone, we can say that on that long bone we have an epiphysis, or 2 epiphyses. These are the ends of our long bone. They have spongy bone that is composed of trabeculae, or these beams of bone that withstand stress, and they're covered with a layer of compact bone, so really thick bone. We'll look at that here in a little bit. The epiphyses are large, and that allows really strong joints, and it allows really good attachment points for tendons and ligaments to help hold those joints together. And then we'll see that the joint surface is covered with articular cartilage, or hyaline cartilage. The diaphysis is the shaft of the bone. It's cylindrical, it's compact bone, it has a central canal inside of it that is full of marrow. On the inside, it's also lined with endosteum, and we'll get into that in a second. So here's what we were just talking about. Here's the epiphysis, the end of that long bone, and here's another one down here. And we have our diaphysis, the shaft of that bone. So inside of those epiphyses, we see spongy bone. Don't let spongy make you think that this bone is soft and squishy. It is just looks like a sponge, and so it is actually made of these really strong trabeculae that help to resist stress in different directions. So as you're jumping up and down or wrestling or whatever it is that you do, those trabeculae are formed to add strength to that bone. On the outside, we have compact bone. And so you can see that on either side of that epiphysis. It wraps all the way around it. In our diaphysis, though, we have really thick compact bone laterally. And in the center of that hollow cylinder is our medullary cavity, and that is going to be filled with marrow. Now, over the ends, highlighted here in blue, on those epiphyses is our articular cartilage, or that hyaline cartilage that is going to reduce friction and offer a little bit of lubrication in those joints. And then on the inside, we have the endosteum, so it's a layer that's going to be lining that medullary cavity. On the outside, which is not labeled here, is the periosteum, and that's a connective tissue layer that helps our tendons and our ligaments fuse into that connective tissue. So again, I just mentioned this, the periosteum. This is what's covering that outer layer of bone, and we see that it's dense irregular connective tissue. So again, we're going to be able to withstand stresses in lots of different directions. You'll be learning a lot about these different cell types in your lectures, but we have two types, osteoblasts, and the blasts are going to build bone, and we have osteoclasts. They're going to cut or chisel away at bone. And so these osteoblasts and osteoclasts can be found below that periosteum, and as we age, the osteoblasts are going to be depositing bone, the osteoclasts are going to be cutting away at bone, and what this is going to do is regulate our blood calcium levels. They're going to remodel our bone as we get older. They might make it wider. And so these two cells are going to be important for you to know. Our osteoblasts, as they're building bone, they actually build bone around themselves, and they get trapped in these little holes called lacunae, and they end up becoming osteocytes. And site means cell, osteo means bone. So bone cells are osteocytes, and then they come from osteoblasts, or bone building cells. And then our osteoclasts come from a different sort of origin, but those are our bone cutters. On the inner surface of our long bones, we see that endosteum, and again, we're going to have some osteoblasts and osteoclasts just below that as well. Now, our compact bone is thicker in the center of the diaphysis. And so think about that for a second. Why would it be thinner around our epiphysis and thicker in the center of that diaphysis? Hopefully you said that it was thicker in the center of that diaphysis because that is going to be where it's going to be experiencing a lot of stress. It needs to be really strong so it can hold up the weight of our bodies and resist those forces. Again, spongy bone is made up of those trabeculae, and you'll see those when you're in your labs. Now, flat bones form a little bit differently than long bones, but just to take a cross section there, we can see that it's almost like a bone sandwich. So they're flat, we have compact bone on the outside, compact bone on the outside, and in between those two layers of compact bone, we have a lot of spongy bone in there. And in that spongy bone, we'll be finding bone marrow. Now, one of the slides you're going to be looking at in lab is called ground bone. Basically, we've taken some bone, we've taken a cross section, ground it down a little bit so that we can expose this structure called an osteon. And right here, as I'm highlighting it, it's this large circle. Up in the top right, you can see an osteon right here in the middle. We have one that is sticking up that has been cut in half so that it's sort of pronounced in this image. But the osteons are basically our modular units of bone, our functional units of bone that are going to build this really strong, compact bone. They are composed of layers called lamellae. And so you can see here in this black and white image, we've got a little bracket, and this line is coming over here to a lamella. And if we trace that lamella around, we can see that this is sort of like the layers of an onion or layers of a tree ring that are in these concentric circles from the inside outwards. And this is going to, again, sort of be how these osteons are built. In the center of those osteons, we have a central canal, and inside of that central canal, we see blood vessels. So you can see some arteries in this image, but you would also have veins and nerves in there as well. The lacunae, I mentioned these earlier. These are these dark spots. This is where we had osteoblasts as they were depositing bone from this deeper lamella right here. They were on the outside of it, putting down bone, and they built bone around themselves, not thinking that they would get trapped. And they said, oh man, I can't go to work anymore. I'm stuck in this little house, and I'm going to be an osteocyte. And these osteocytes then are essentially regulating and monitoring our bone matrix. Now, these osteocytes need to communicate with each other, and they need to receive nutrients from the central canal. So there are thin little tubes called canaliculi. They look like sort of the spider legs coming off of our lacunae that actually reach out to other osteocytes and lacunae and are going to allow nutrients and wastes to diffuse in and out and communicate with other osteocytes. Now, another slide you'll be looking at is called the decalcified bone slide. In here, what is really nice that you can see is our bone marrow inside of that spongy bone. We can see this layer of compact bone out here. You can see an osteocyte there, but on the inside, between that bone marrow or that spongy bone and our compact bone is where we're going to see that endosteum, and then on the outside we're going to see that periosteum. Now all the way over to the right you can see some skeletal muscle. That skeletal muscle is going to be adhering to that periosteum at those points of connection. So thanks for your attention today. I hope that we didn't go too quick through that, but make sure you're practicing your layers. Get used to all those bones. You will be expected to identify some bones on your next test, and you will be looking at some of the microscopes and having to identify osteons and the various structures associated with them. Have a great afternoon or morning, and we'll see you next week.